Hello everyone, my name is Heriberto Moran, and the title of my talk is going to be on the understanding the control of HDM2 and synthesis and its contribution to cancer development. So why I'm interested in this topic is because HDM2 it's an oncogene whose due to its critical role in tumor development, understanding the mechanism by which HDM2 expression is controlled, this led me to investigate whether HNRMPA1 plays a role in the HDM2 gene expression regulation. But before I discuss my results with you, I would like to go over some background information of what are oncogenes, tumor suppressors. So, oncogenes are a group of genes implicated in the development of cancer, and HDM2 is an example of an oncogene. So there are three types of genes that are associated with cancer. The first one is called the proto-oncogenes, and these proto-oncogenes produces protein products that normally enhance cell division or inhibit normal cell death. So the mutated form of these genes are called oncogenes. The second group of genes who are associated with tumor development are called tumor suppressors, and this group of genes produce protein products whose function is to prevent the cell division or cause cell death. And one of an example of the tumor suppressor will be the p53, and p53 it's regulated by HDM2. And the third group of genes that are associated with tumor development are called DNA repair genes, which help prevent mutations that lead to cancer. So proto-oncogenes and tumor suppressor genes work much like the accelerator and brakes of a car, respectively. So as you know, let's say the normal speed of a car can be maintained by the controlled use of both the accelerator and the brake. Similarly, controlled cell growth is maintained by regulation of proto-oncogenes, which accelerate growth, and tumor suppressors, on the other hand, tumor suppressor genes, which slow the cell growth. So any mutations that produce oncogenes will accelerate growth, while those that affect tumor suppressors prevent the normal inhibition of growth. In either case, uncontrolled cell growth occurs. So there is a correlation between HDM2 overexpression or overproduction with an increase in tumorigenic potential. So as I mentioned before, understanding how the production of HDM2 is regulated will provide us with a useful insight into the mechanism of cancer development. We can use this information to develop new targets for HDM2 for cancer therapeutics. So what is HDM2? HDM2 is an example of an oncogene whose main function is in the regulation of 53. And what does HDM2 stands for? HDM2 stands for the human form of the double-minute amplified. So what is HDM2? It's an oncoprotein. It's a key regulator of a variety of fundamental cellular processes, originally identified as being amplified on double-minute chromosomes in transformed mouse fibroblasts. So right after this it was discovered, after its discovery, it was found to be a negative regulator of the tumor suppressor p53. And we have evidence that HNRMPA1 participates in the regulation of mRNA nucleocytoplasmic trafficking. So what do I mean by this? It participates in the export and of certain messenger RNAs that code for proteins, and we have evidence that HDM2, the HNRMPA1 binds to the messenger RNA of HDM2. So what is HNRMPA1? It's an RNA binding protein with many important cellular and participates in the control of various cellular processes such as RNA splicing, also the export of certain mRNAs, and it consists of it has this modular structure consisting of one or more RNA binding domains, and other it has other so this is the regions where RNA is going to bind to, where this is the location where where HTM2 will bind to, so I would like to share three results that I was excited about my project. So we hypothesized that that the treatment of young IMR90 cells, which are the fibroblast cells that we use to study this to complete this project, when we treated these cells with actinomycin D, which is an inhibitor of transcription, we hypothesized that this should lower the HTM2 protein levels because of the accumulation of A1 in the cytoplasm. And indeed, we find that when we treated the cells with this inhibitor, we saw a slight decrease in HTM2 protein levels, which correlate with the cytoplasmic accumulation of A1 during the actinomycin D treatment. Although this result suggests that cytoplasmic A1 could be reducing the translation of HTM2, further studies are needed to confirm this finding. We also hypothesized that the expression of a mutant form of A1 will interfere with the nucleocytoplasmic shuttling activity of wild-type A1, as previously demonstrated. So in order to investigate the effect of this plasmid, which retains so this plasmid will be in the nucleus, so it's a nucleus localized plasmid, which will prevent the nucleocytoplasmic shuttling of A1, and as a result of this, it will, if it binds to HTM2, it will prevent the export of HTM2 from the nucleus to the cytoplasm. So when we transfected these cells with this plasmid, we find that HTM2 was not exported to the cytoplasm. We provided this with results with using real-time PCR and also semi-quantitative PCR. We demonstrated that this in fact was the case. We repeated the experiment 3 times, and we found that that the HTM2 mRNA was no longer found in the cytoplasmic fraction of the cells that were transfected with this plasmid. The third result that I was really excited about was the fact that we performed RNA protein interaction studies, and we found that so we got this plasmid from HTM2, and we used certain fragments of the HTM2 mRNA, and we did RNA electromobility shift assays, which is a type of technique that is used to provide conduct studies that will indicate whether HTM2 mRNA is binding to the protein, to the HNRPA1 protein, and we found that when we did these studies, we found that A1 was identified as the RNA binding protein to directly associate with the biatylenated HTM2 mRNA. So the direct association between A1 and biatylenated HTM2 mRNA appears to be specific, since these interactions were not observed with similar RNA binding proteins such as HNRPA0. So further studies, furthermore, association between A1 and negative control biatylenated beta-actin were not observed. So these results demonstrate that A1 has specific binding to biatylenated HTM2 mRNA. In conclusion, our data demonstrates that HNRPA1 is a new binding partner and plays a key role in the regulation of HTM2 synthesis. And discovering new binding partners for HTM2 will enable us to develop new treatments for cancer. Thank you.